Assalamualaikum everyone, good day. Don't know when you'll be watching this. So welcome back to essentially the third lecture for part 2 for immunology. And in the previous 2 lectures we talked about immunological memory and how we use that to develop vaccines. And then we talked about different infections and how the immune system responds to them. And today we're going to talk about immunodeficiencies, and one of the most famous types of immunodeficiencies, as you may know, is caused by the virus, human immunodeficiency virus, which also causes the acquired immune deficiency syndrome, AIDS. So let's get to it. So at the end of this lecture, I hope you can distinguish the difference between primary and secondary immunodeficiency, as well as some features about these. And then we're going to go a lot more deeply into HIV as the cause of AIDS, including a bit about the HIV life cycle and how it actually progressively suppresses our immune system. Gut leakage is an important element of an HIV infection, and then how AIDS develops. And then we'll touch briefly about the progress and challenges in controlling HIV/AIDS. As you know, HIV/AIDS is also a pandemic. Before we had this COVID-19, I think, and before the last pandemic, which was the H1N1, HIV/AIDS was the one that was declared to be a pandemic. So it's certainly still a big problem in many parts of the world. So let's talk about immunodeficiency first. What is immunodeficiency? It's basically when our immune system is unable to protect the host from disease-causing agents. So you know the primary purpose of the immune system is to defend us, but when there are deficiencies in that system, it cannot do its job, and then you are now considered immune deficient. And there are 2 main types, which is primary immunodeficiency or secondary or acquired immunodeficiency. With primary ID, it's usually because of a genetic underlying condition that is inherited or a developmental defect. So it happens much earlier, probably even before they were born, and they are born with these deficiencies. The second type, which is acquired after birth, and that can be through things like unsafe injections or mother-to-child transmission of HIV, for example, or malnutrition or other chronic diseases. So these are all things that can impact the immune system and cause people to become immune deficient, even though they don't have any genetic underlying immunodeficiencies. So let's talk about primary immunodeficiency first. It's going to be basically a shorter part of the lecture. The most severe form of this type of inherited immunodeficiency is a group of disorders called combined immunodeficiencies, or CIDs. And this is when you actually have complete absence of T cells or very, very impaired T cell function. And this is also often accompanied by some problems in your B cells and antibody responses as well. But largely you've got, in terms of the distribution, what is more common is the B cell types of deficiencies, but the most severe kinds is when you have T cell types of deficiencies. So this is just a figure to show you how it arises, and usually it's defects in how in the stem cell development, and it prevents the development of your lymphoid cells or the progenitors to your neutrophils and your monocytes. And the most extreme form of CID is called SCID, or severe combined immunodeficiency, and this is when you actually don't have T cells, or your stem cells for the lymphoid lineage is also completely gone. That means you don't have both, you don't have your lymphocytes, so that's a very key part of the immune system, as we know. And so what happens is you'll have defects in how your cytokine signaling goes. You might have defective, so these are the things that actually may have caused the defects. So it's either the defects in cytokine signaling, first and foremost, or your lymphoid lineage suffers premature death, maybe because of some toxic metabolite accumulation, or you've got some genetic rearrangement problems, or there's any disruption in the TCR signaling during development. So you can lose T cells, or both T cells and B cells. Now what happens once you do have SCID is you get obviously very, very terrible symptoms when you get infected with anything. And you'll start detecting this in children very early once they're born. They'll just keep on getting sick, and they'll have a lot of infections, even very minor infections for a lot of people can become quite deadly for babies who have SCID. And there is a screening test available to tell parents if their child might have SCID. And what they do is they just take a little bit of blood from the baby's heel, and then they will do a PCR. And it will tell you whether or not this child has SCID. And after that, it's really, really aggressive treatment. In fact, some of these children will have to live essentially in a bubble environment to prevent them from getting infected with anything. So this is a little picture of, I guess, a baby and their sibling separated by a bubble. So yeah, your immune system is so crucial in helping you survive in this very harsh world. But as we talked about, and I thank a lot of you for responding to the forums, sometimes that immune system also becomes the cause of our misery when there's any overinflammation. But by and large, you wouldn't be able to live very long because children who have SCID often do die within 1 to 2 years because their system cannot cope with that constant recurring infections. So there are other combined immunodeficiencies as well. One is called the bare lymphocyte syndrome. So as the name suggests, the lymphocyte is bare, specifically bare of MHC molecules, and this obviously makes it very difficult for them to undergo cell-mediated immunity because the MHC class is very important for antigen processing. The other type of syndrome is called Deficiency syndrome, and because of deletions in a region on chromosome 22, we have reduced T cell numbers under this condition. But if it's not a very severe form of this condition, it can be managed with antibiotics during bacterial infection, and people can still, I think, have relatively normal lives. Then you've got what's called Eldridge syndrome, and what happens is in these people there's an X-linked gene deficiency, and they have less than normal levels of IgM. So they'll get recurrent bacterial infections as a result of this. So there's some pictures of the clinical features. So another type of immunodeficiency, as I mentioned, is a very common type, which is the B-cell immunodeficiency, typically the less severe kind. So it's quite diverse, and it can generally be because of an absence of mature recirculating B cells, plasma cells, or antibodies, or selective absence of antibody classes. So what happens is you would get some recurrent bacterial infections. So in X-linked Hypogammaglobulinemia, you'll have extremely low IgG antibodies and basically no other antibodies. So that's managed by getting a passive administration of antibodies. So I think you may know in some patients, or actually a lot of patients in Malaysia right now who are having COVID-19, one of the treatments that they're trying out is giving people a convalescent plasma. What that means is somebody gets COVID-19, recovers, they donate their blood, and their blood or their plasma, which contains the antibodies, will be donated to the person who is currently going through the infection, and that seems to help patients recover. And that is a form of passive administration of antibodies. And this is a very common technique in treating other infectious diseases like rabies as well. Common variable immunodeficiency disorders is when you have less levels of one or more antibody isotypes or classes and an impaired B-cell response to antigens. But by and large, the numbers are normal. It's just their capability to respond to antigens and make antibodies is impaired. And then the last one is selective IgA deficiency. So IgA is actually one of my favorite isotypes, because that was what my PhD was about. And it was very annoying because there is this very well-known selective IgA deficiency in the population. And it affects actually quite a number of people. I think there's some estimates that 1 in 400, 1 in 700. So every now and then, when you run a test to see IgA response to an antigen, there will be that one person in that sample or in that study population that will have no IgA, and it's always like really like interesting. So IgA deficiency tends to not be very impactful in terms of a person's health condition, because, as you know, you've got like IgM, which is also an acute acute phase antibody, and all the other subclasses are able to sort of top up. IgA is really important though for your gut lining and mucosal lining, because that's the secreted type of antibody, but you also have some levels of secreted IgM, I think. So you've got slightly normal, greater than normal susceptibility to respiratory and gut infections, but nothing too severe. Right, so you also have some immunodeficiencies with your innate immunity. It can be because of problems in the myeloid cell lineage or in the complement system, so you get less numbers of phagocytic cells. And this can manifest at a few levels. Either the cell motility is affected, the adherence to and phagocytosis of organisms might be affected, or they might affect the intracellular killing process. So, for example, in terms of the motility or the adherence, you might have leukocyte adhesion deficiency because of a dysfunction of the integrin adhesion molecules in a defect that's localized to the CD18. So what happens is basically the leukocytes cannot adhere, finish their rolling, and then extravate. That's an important process for them to actually get through and get to the source of the infection. So this makes a person who has LAD more susceptible to certain gram-positive, gram-negative bacteria infections. So just because I don't have CD18 means that I can't hold on? Precisely. Another type is a chronic granulomatous disease. So you may have heard of this disease before, CGD, and this is because of defect in the NADPH oxidative pathway. You can recall back in your biochemistry or even in your general microbiology. So this is an important pathway that produces the superoxide radicals and other toxic species that's usually used to kill any pathogen that's been phagocytosed. So it's gobbled up, and then these superoxides will then kill whatever has been gobbled up. But when that process is deficient, it obviously doesn't get killed. And if you remember from our last lecture, what happens when you can't kill something that's been ingested or something that is now intracellular? Sometimes you have to form these granulomas to just prevent them from spreading. So a characteristic feature of CGD is the production of formation of granulomas, so these small mass of inflamed tissue. Finally, I think the last bit about innate immune deficiencies is complement deficiencies. So this results from defects in the complement system, and it can lead to generalized failure to activate the complement, or actually failure in different pathways, and it will result in alternative pathway activation. And what happens is people who have these deficiencies will have increased susceptibility to bacterial infections or immune complex diseases. So for example, in people who have deficiency in properdin, which stabilizes the C3 Convertase, they will have increased risk of Neisseria infection. And people who have defects in mannose-binding lectin will have increased susceptibility to a range of infections by bacteria or fungal agents. Right, so how do we treat these primary immunodeficiencies? For the more severe kinds, I think what they're looking into is the potential use of gene therapy. So you try to identify the gene and then you replace it or you could actually skip one step down and replace the missing protein. And this is, as we discussed just now, passive administration of immunoglobulins. That's an example of replacing a missing protein, or giving the recombinant interferon gamma, for example, or you replace the missing cell type or the lineage. And there has been some success in HLA identical donors for certain types of cells. All right, so, sorry. Yeah, I think there should be another one where it says the other way you can treat it is by aggressively, oh no, sorry, we'll move on. So next we have acquired immunodeficiency. So with acquired immunodeficiency, you're not born with it, obviously, and so how do you actually get it? And what kinds do you have? So besides HIV, which we'll go into greater detail in a bit, you've got the kind called hypogammaglobulinemia, which is similar to primary immunodeficiency, except that you don't have any evidence that it was inherited. So not the most convincing, but that's what we know about this hypogammaglobulinemia for now. Also agent-induced immunodeficiency. So this can be exposure to environmental agents. These can be drugs that we take that suppress our immune system, or radiation, for example. So people who are undergo cancer treatments often also have some immune suppression. And then, of course, a very understated cause of immunodeficiency is severe malnutrition and micronutrient deficiencies. So if you don't have enough nutrients for you to build your immune system, then you will have a defective immune system. And unfortunately, this is a really main problem in a lot of places that have poverty. So the children often tend to be more vulnerable to infectious diseases because they're also malnourished, so their immune system is less robust. Okay, so let's go into the titular part of the lecture, which is HIV/AIDS. So HIV was first recognized as an opportunistic infection in a cluster of individuals in the US. And what they first noticed was there was something that was going around infecting mainly homosexual men, or men who have sex with men. And they were having very odd infections, like opportunistic fungal infections. So remember I told you fungal infections are not very common unless you are immunocompromised. So these group of people had these opportunistic fungal infections. And they also had a very rare skin tumor called Kaposi's sarcoma. And then it was later determined that it's because they had very, very decreased levels of CD4 T cells, which is caused by a particular virus that is later now known to us as human immunodeficiency virus, or HIV-1. HIV-1 and 2, there can be both. So the HIV is a retrovirus from the lentivirus genus. And the way you get HIV is basically through intimate contact and infected bodily fluids. So these can be through having sex without a condom, so never do that. It could be passed from mother to baby. It could be sharing, injecting equipment. So please don't do this either. And then contaminated blood transfusions and organ transplants. In a lot of places, we have very strict blood safety regulations, but depending on where you are and which part of the world, some places still do not have as high standards for blood safety. So be very, very cautious when you are in those areas. Right, so besides these routes, there is no other route that we know of that you can get HIV. So what happens when you get infected with HIV? So the HIV will try to complete its life cycle. So it carries 3 structural genes, the GAG, POL, and the ENV, and the 6 regulatory or accessory genes, TAC, REV, NEF, VIF, VPR, and VPU. So the HIV actually very cleverly will infect cells that carry the CD4 antigen on their surface. And as we know, one of the key cells that carries the CD4 is the T helper cells, but also other immune cells like monocytes and macrophages. So there's a very high affinity between the GP120 of the virus and the CD4 molecule. But there are also 2 known co-receptors for HIV, which is the CCR5 and the CXCR4. So together with the CD4, they somehow allow HIV to enter mostly our CD4 T cells. So this, I'm not going to go into great detail. You can have a look at how this actually works. But with most viruses, it's all about getting first that binding to the right receptor, and then being able to fuse and enter, having the genetic material be integrated, the provirus, and then they will start hijacking the host protein machinery to make more parts of itself, and then they will start releasing new viral particles that have matured. So this is quite a very scary image. This is an infected T cell, and you can actually see the viral particles being released out of a dying T cell. So the progression for HIV immune impairment is basically first during the acute phase. Acute phase, obviously, when you first get infected, you don't actually see any of the antibodies, but the HIV infection is spreading and the viral load is quite high. Then you go into this asymptomatic period where your CD4 cell starts to decline. Obviously, they're dying because they're getting infected with the virus, but you can't really feel any symptoms yet. And this is because there's sort of like a check and balance between the CD8 T cells that keeps the viral application low, but in some cases the viral application can still continue, and as the CD4 cells keep falling, then suddenly these can progress into AIDS, especially without treatment, and that will start letting you have these other infections. So it's not the HIV that will kill a person. It's all these secondary infections or opportunistic infections that start infecting a person because the CD4 T cell levels have gone so low. So this is the course of an HIV infection. You've got on the y-axis the CD4 T cell count in the blood, and then on the x-axis you've got the duration. And basically this is the acute phase that we talked about. The CD4 T cells starting to decline, but still quite reasonable amount. Not noticeable, you don't see any symptoms yet. And the viral load in the blood, which is on the second y-axis, is quite high. When you enter the asymptomatic phase, the number starts dropping, and the CD4 T cell number is also gradually declining, but you're also still not seeing any symptoms yet. Then once you start seeing a further reduction in the CD4 T cells, that's when you enter the AIDS phase, and that's when the virus levels shoot up, and you also start getting all the secondary infections. So what happens, basically, what level do we consider to be the level of CD4 T cells that makes somebody enter an AIDS phase? This is stage 3. It's usually when your CD4 T cell count is less than 200 cells per microliter, or it's less than 14%. And what you will start seeing is the presence of AIDS-defining conditions. This list, very tiny font here, is the list of AIDS-defining conditions, but here you've got it, basically the main ones. You've got these pneumocystic pneumonia, you've got NTB. TB is an AIDS-defining illness. You've got invasive cancer, Kaposi sarcoma, one of the early ones that they noticed. So what you want to do with the patient is you want them to avoid from getting in this stage. So not everybody needs to get treated when they have HIV, but they will start getting you on treatment once they notice your CD4 T cells start dropping, especially below the 200 or 250. And previously how they would do this is they would need to use a flow cytometer to measure the CD4 T cells. But last couple of years, there's a lateral flow test that doesn't require any equipment. It actually gives you a cutoff of 350 and below. This is actually the new WHO guidelines, where they want you to start treating people when they have 350 cells and below. And so this test is kind of semi-quantitative. It gives you an indication whether or not somebody's above or below the cutoff value. And I'm very proud to say this was developed in my old lab when I was doing my PhD. And I remember it was a lot of struggle getting this test to work, because it's quite a new type of test where you trying to make it semi-quantitative instead of just qualitative yes or no. And now I think they're getting some good results, and it's been approved for regular use. So we'll talk a bit more about the importance of having very simple tests like this, because it means people living in areas without labs can get tested and then hopefully can get treated. Right, so the progression to AIDS and gut leakage. Interestingly, it's actually hard to find any infected CD4 cells in the circulation, because once they get infected, they are killed very rapidly. So in less than 1.5 days. And any of the infected cells may actually be localized to the GI tract. So it's very hard for you to actually find viral CD4 cells with the virus in the blood. But what you would start seeing as you go into the AIDS phase is that there's a significant depletion of lymphoid tissue and CD4 cells from the GI tract. The Th17 cells, which expresses CCR5 and CXCR4, are the primary targets. And these cells are actually present in the gut to regulate a lot of the innate and adaptive responses to the gut microflora. So as we know, our body is host to a lot of different kinds of microorganisms, many of them good friends of ours, and some of them, we don't mind them there. But that's because we're able to kind of keep them in check with our immune system, and also just their own communities are able to prevent one single microbe kind of getting dominant and colonizing the whole thing. But when you have the HIV attacking the gut, specifically these Th17 cells that regulate this, obviously you start getting some problems in the gut. And the main thing is actually you do have a physical disruption of that barrier. So we're happy to have all these organisms outside, but we don't want them entering, obviously. But because of the HIV infection, you start getting what we call gut leakage. The gut starts to leak. So you start having this a translocation of the microbes or the microbial products across the epithelial lining. And what that does is it actually stimulates the immune system. It actually brings in more CD4 T cells for the virus, for HIV, to infect. So very clever, very destructive. This is how you would see a patient, this is a person who doesn't have HIV, so you can see these are the, what we call, I think, the patches of lymphoid cells, which is composed of a lot of CD4 T cells. And then this is in the gut of a person who's HIV positive, so clean of these aggregates. And even under histology, you can see all the brown staining is the T cells, and here it's very, very sparse. And this one is the percentage of the T cells comparing different locations of HIV positive and HIV negative individuals. You basically have a lot less T cells across these different locations for HIV positive patients. So in other immunologic symptoms that comes with AIDS is that you actually have a decrease or absence of delayed type hypersensitivity to antigens. You might also have decreased serum immunoglobulins or impaired cellular responses to antigens. You also get impairment in natural killer and dendritic cells, so it's really becomes a kind of a combined immunodeficiency. And you might even see some dementia because HIV has actually been detected in the nervous system as well. So this kind of shows you some other immunologic abnormalities which we touched on. One thing quite interesting is, for example, in the antibody production, early in the infection, you might actually have more antibodies being produced, but they're very non-specific. And then in the late stage, then that's when you start having less antibodies for these patients. Same with the cytokine production. You have increased levels of some cytokines early on, and then later on there's a shift from Th1 to Th2. And often, as we know, the Th2 subtype is basically the less protective kind. Right. So just now I mentioned about point of care diagnostics, and it's a very important, I think, turn of tide for HIV on top of finding the right antiretrovirals. But one of the main problems previously was diagnosing HIV was quite costly and also accessible in the places that it was most prevalent, and that's in sub-Saharan Africa. Once you had a new type of test that was very simple, very cheap, that you can run outside of the lab, you had at least 27% of people were able to find out their results. So that actually, and it's especially useful for pregnant women. And what that does is once they know that they are HIV positive, they start taking the antiretroviral treatment. So it reduces their risk of transferring their HIV to the baby when they give birth. It also allowed for people to kind of get more. I mean, there's more people who can offer giving the test now. You don't just have to go to a central hospital. I think one of the problems you realize now with the COVID-19 is we only can rely on the PCR method, which is obviously costly and very, very lab limited. You can only do it in a lab, and not only that, you've only got certain labs that can actually do it. So something simple like this is what you call a real game changer in how you control an infectious disease. So because of that, and coupled with ART, you're able to get people who to know that they have the infection, they get access to the drugs, and then the incidence drops down and the number of deaths also drop down. It's really transformed the epidemic. I think when people find out, even if they don't need treatment, for example, they have the responsibility to change their behavior as well, and to make sure other people don't get infected. So it's been a very important part of the control. And ART especially, basically a lot of people are able to grow old with HIV, whereas previously it was a death sentence. If you get HIV, it doesn't take very long to progress to AIDS. So the HIV drug targets that's used is basically antiretroviral drugs that either antagonize the receptor. So this could be something that antagonizes CCR5, for example, or drugs that inhibit the fusion of the virus, or it actually inhibits the reverse transcription of the virus, or it inhibits the integrase, or it inhibits the protease, which allows the virus to cleave off. So there are different points in the life cycle that the antiretroviral drugs can target. But one thing we definitely don't have for HIV until today is a vaccine. So one of the reasons, because HIV mutates rapidly, so it's very difficult to design a vaccine that can protect people against HIV. But a lot of viruses mutate rapidly. So we know flu mutates very rapidly, but we have a vaccine for flu. But I think the bigger problem is that there is less understanding about what is actually going to protect somebody from disease. So we don't know, for example, if just having this antibodies alone is likely to block infection. And if you remember, you can either have the live or the non-live vaccines, and that will induce a certain type of immunity. In any case, there are different things that people have been trying to do. I think the DNA one is the one that people are trying to get to work as much as possible. The whole inactivated, live attenuated synthetic peptides, recombinant bacterial vectors. So none of that has worked so far. But just to give context, for typhoid it took 100 years to make a vaccine. For pertussis it took 89 years. For polio it took 47 years. For measles it took 42 years. HPV 33. They didn't put TB here. Probably longer, because TB is a lot older. And HIV still, I guess, coming up to 30 years or longer now. And I think a lot of people are banking on a COVID-19 vaccine soon, but this type of graph is always going to be a bit of a reality check. Hopefully it does come, but for now I think we do need to change the way we live for a while. So the recent strategies for HIV vaccine is the one that wants to elicit both humoral and cellular immunity. So obviously as we know, that usually requires the use of a live vaccine, but you could also aim for a DNA vaccine, which I mentioned. So it's now still being tested. Yeah, I need to check again what's the latest, but as far as I know, there haven't been any breakthroughs yet with HIV vaccine. Now, an interesting controversy which happened just last year is this idea of gene editing to prevent HIV. So using the CRISPR-Cas9 system, some scientists in China claimed that they germline edited these babies so that they are less likely to get infected with the HIV virus. So we don't know the state of the children right now. Obviously this is unethical because it was decided that germline editing could not be ethical because you cannot get consent from unborn children. You can only get consent technically from their parents, but their parents do not have that level of right to determine a person's genes. So the rationale goes. So this was something that was very, very controversial. So what are some persistent challenges in HIV control? So we talked about the HIV-TB co-infection. That's a huge problem. So people are now surviving with HIV much longer, but then if they get TB, that's usually what kills them. So it accounts for 25% of AIDS deaths, or 40% in Asia. But it's not just TB, there's also HIV, hepatitis C virus co-infections. And this is partly because they share the same route of infection or transmission, so based on bodily fluids. So you get a lot of people who inject drugs getting hepatitis C virus infections, and then they can also get HIV that way. So they get both, and having both together makes it worse. Then you've got HIV and cryptococcal meningitis. So all these co-infections these are the ones that are really challenging control. Okay, so that's the end of this lecture. I'll see you in the next one.